Hello everyone, I am Blessy Antony, a third year PhD student in the Department of Computer Science at Virginia Tech. Today I'm going to present on my current research project titled Zoonosis Prediction Using Language Models. Zoonosis is an infectious disease that has jumped from animals to humans. They represent a major public health problem around the world due to our close relationship with animals in agriculture, as companions, and in the natural environment. As of April 2019, there were 1.6 million known animal viruses in nature, but only 219 viruses have been documented to infect humans. Some well-known examples are H5N1 avian influenza and Ebola virus disease. We can now add the latest SARS-CoV-2 virus that caused the COVID-19 pandemic. With only 0.01% of the animal viruses known to infect humans, the sense of mystery is what causes a virus to be zoonotic. Scientists have been studying which mutations confer a virus the ability to switch hosts and adapt to a new host. For example, the zoonotic and highly pathogenic H5N1 avian influenza virus can be traced back to the 1997 H5N1 poultry outbreak in Hong Kong. This virus had a PB2 polymerase subunit protein that binds with the ANP32A transcription factor in the host for virus replication. This virus required a mutation in its PB2 polymerase subunit protein in order to be compatible with the human version of the ANP32A. Without this mutation, this human transcriptional activator provided a potent block to AIV replication in humans. Similarly, we have the examples of simian immunodeficiency virus from chimpanzees that had mutations in the VIF and VPU genes, while the coronaviruses had mutations in the spike protein for adaptations to the human host. Thus, mutations in a virus enable them to switch hosts, evade the immune system, and infect, adapt, and replicate in the new host. Our long-term goal is to computationally predict the mutations that enable an animal virus to shift hosts and adapt to human cells. We are breaking this challenging zoonotic prediction problem into two smaller and hopefully tractable questions. First, given the protein sequence of a virus, predict which hosts the virus will infect. We can then take a step further to ask the question, given the sequences of a virus that infects both animals and humans, determine which mutations may cause the host shift. So we intend to develop machine learning models to identify one or more viral genetic elements responsible for species jumping and adaptation in human cells if there are any. We hope that the machine learning models we develop to answer the first question may then help in pinpointing the mutations for the second question. In this talk today, we are going to focus on the first task of host prediction. The studies done so far try to answer the question of predicting the host of a given viral sequence while either focusing on one specific protein or a specific virus of interest or both. Further, these studies only go on to answer the binary classification question of whether the virus from which the sequence was sampled will infect a human or no. Thus, the multi-class classification challenge still remains unsolved, that is, to identify hosts of an arbitrary protein sequence from arbitrary viruses. Here's our approach to tackle the task of host prediction. The primary structure of proteins is sequences. We can consider the chain of amino acids to be a sequence of tokens with each amino acid representing a token. So we used language models based on the analogy that the protein sequences follow grammatical rules like the natural languages that we use for our day-to-day conversations. So we can train natural language processing models such as transformers to learn the amino acid sequence language. For this host prediction task, our input is a protein sequence sampled from a virus. The language model we have used is derived from the transformer, which contains the self-attention. We use the encoder portion of the model to learn embeddings for the protein sequences, followed by a multi-layer perceptron for the multi-classification problem. The output is the predicted probabilities for the host of the virus from which the given sequence was sampled. Some implementation details are: we fit the model for the multi-class classification problem using focal loss. We split the dataset in the ratio of 80-20 for training and testing datasets. And finally, we repeated all experiments 5 times for generalizability. Our dataset is from UniProt, which provides protein clusters called UniRef90. These are groups of protein sequences with at least 90% sequence similarity, and each cluster is represented by one representative protein sequence. We downloaded these representative protein sequences of virus infecting mammals or aves and filtered for protein sequences from viruses infecting more than one host. The final dataset comprised of approximately 19,000 sequences from 97 viruses sampled from 59 hosts. This is a highly imbalanced dataset with almost 80% of the sequences coming from humans. Finally, we used the sequences pertaining to hosts with at least 1% representation in the dataset, which gave 8 unique virus hosts. We will now look at the results of host prediction. Along the x-axis, we have the different models that we compared. We configured the deep learning model such that the complexity of the models in terms of parameters is similar, with each of them having roughly 12 to 14 million parameters. Along the y-axis, we are looking at the distribution of the macro AUPRC, that is, average of the AUPRC computed for each class over the 5 iterations. The logistic regression and random forest models performed close to 0.5, and the fully connected neural network model also had similar performance. The recurrent neural network, however, performed the worst due to the vanishing gradient problem known to be prevalent in RNNs. The long short-term memory model performed better due to its memory cell. And finally, transformers performed the best, which is not really surprising given that it is empowered with self-attention. Here we are looking at the distribution of the AUPRC scores for each host class over the 5 iterations. We observe that the model has difficulty in learning about classes of protein sequences that are less represented in the dataset. We also see that the transformer model classifies the human host close to perfection. We now move on to the second question of zoonosis prediction, that is identifying the mutations that cause the host shift. We are in the midst of looking at different ways to interpret the predictions of the transformer model. We started with looking at the average of the self-attention values of each amino acid token in the sequence across all positions. Here is the analysis of the attention values of one spike protein sequence from SARS-CoV-2 virus infecting humans. The functionality of the 3 of the top 10 positions with the highest attention values have been determined as the receptor binding domain. This domain is known to comprise of roughly 12% of the whole sequence. That is, these positions of the spike protein are involved in the binding with the human ACE2 receptor, thereby enabling the virus to infect the humans. Here, we see a low-dimensional t-SNE visualization of the embedding learned by the transformer model for each protein sequence. The different colors indicate the different host classes. We can see a fairly distinct cluster for human on the left-hand side. While the right-hand side also has distinct clusters for different classes, there is an overlap between the different classes, thereby potentially indicating the presence of zoonotic sequences. So currently, we are using saliency maps for interpreting transformer models to identify important amino acids for host prediction. Further, we also intend to use pre-trained models to learn embeddings of protein sequences. And we also plan to leverage the structural information of proteins for this zoonosis prediction challenge. I would like to thank the funding sources and Virginia Tech for enabling this research. Thank you.